Okay, we are going to start talking about metabolism. And the first type of metabolism that we're going to spend quite a lot of time on is the metabolism of carbohydrates, and the most famous carbohydrate being glucose. So we're going to look into carbohydrates in general first as a biomolecule, seeing how what they're composed of chemically, and then think about their different functions. So we're going to go into basic chemistry of the monomers in this video and how they can form rings. And the next video we'll talk about how they can be joined into polymers and what those polymers look like. And then the final video on this introduction to the metabolism of carbohydrates, we'll talk about how carbohydrates can function in tandem with proteins. So let's start by looking at the basic chemistry of carbohydrates. They're called carbohydrates because they've got carbon in them and they've got water in them, and that's at that ratio of CHO of 1 to 2 to 1 that we think about carbohydrates. They can be in aldehyde form, aldoses, or ketone form, ketoses, and there's a lot of stereochemistry associated with carbohydrates. There's a lot of chiral carbons in most carbohydrates that we think about, and different stereoisomers possible. And so we'll think about epimers, which are different stereoisomers of carbohydrates and how they function. And carbohydrates fit the theme of a lot of biomolecules in that they are present in monomers, and those monomers can polymerize together to form polymers, just like we thought about with amino acids polymerizing to form proteins, just like we think about nucleic acids polymerizing to form even things as big as chromosomes. Carbohydrates are similar with some important differences. Okay, so first, let's look at some carbohydrates here. Glyceraldehyde is a carbohydrate. Dihydroxyacetone is a carbohydrate. We're going to see these two molecules really soon when we talk about glycolysis, because glucose is broken down into these two molecules, which are converted back and forth to each other. But here's the king, right? Glucose. We think about glucose as one of the main sugar sources for metabolism. And all of these sugars that we have here on this slide are carbohydrates, right? They've got a carbon backbone with alcohol groups peppered all over them. And if you look at one of these sort of carbons in the middle here, you can see why they're carbohydrates. Here's the carbon. Here's H2O, right? So here's glucose, fructose, ribose, right, and deoxyribose, the sugars in our nucleic acids. Glucose and fructose are 6 carbons. The ribose and deoxyribose are 5 carbons. But we can also split the categorization of these sugars into aldoses versus ketoses while they're in this linear form. And obviously, if you've got an aldehyde here, like the left panel, you're considered an aldose. If you have an internal carbonyl group, like a ketone, you're a ketose. Now, if you look at particular carbohydrates, there's a lot of chiral carbons in them, right? And here's mannose, right? There's prochirality at this aldehyde here, the carbonyl group, but at carbon 2, 3, 4, and 5, all four of these carbons are chiral, and therefore have different R or S versions of them. Now, when we have different sugars that differ only by the stereochemistry at one chiral center, we call those epimers of each other, right? And so glucose has epimers of mannose and galactose. They're all epimers of each other because they have the same carbon backbone. They're both aldoses, and they just differ by the stereochemistry at one of the carbons, carbon 2, carbon 4. The other thing to note on this slide is how we're numbering these carbons. Usually we will start, we start at the end of the carbon chain, and with things like aldoses, the carbon number 1 is going to be the carbonyl carbon, right? And that's going to be important later as we think about these linear carbohydrates cyclizing. So let's talk about this. Turns out glucose in your bloodstream isn't doesn't exist at very high concentration as a linear chain. Most of the glucose in your blood is cyclic. And so how do these sugars cyclize? Well, here we have glucose here, and it forms a 6 atom chain, right, a hexagon here. But it turns out that one of the atoms in the hexagon is bridging oxygen. That bridging oxygen comes from, in this case, the 5th carbon. And we think of alcohol groups all the time as being potentially nucleophilic because there are lone pairs, which I'm trying to draw in here, on the oxygen of all of these alcohols. Now, since these lone pairs make the alcohols Lewis bases and nucleophiles, they can attack the partial positive charge, right, on the carbonyl carbon. So this is supposed to be partial positive because of its double bond to the electronegative oxygen. And really, it's this sort of intracellular, or not intramolecular reaction that occurs between the nucleophile and the electrophile that causes the cyclization to occur. Here's a better picture of this alcohol attacking that carbonyl carbon. And this alcohol can attack from the top or the bottom of the carbonyl. And that leads to 2 different stereoisomers of the cyclic compound. Now, either this alcohol oxygen, which is going to become this bridging oxygen right here, is going to attack from the top, pushing the electrons out on the carbonyl to become protonated to form an alcohol. And if this alcohol attacks on the top, then this alcohol will form on the bottom, like we see on the left here, alpha glucopyranose. Or this alcohol could attack from underneath, the bottom, pushing this alcohol group at the top. And that's what we see with this beta glucopyranose. Now, these 2 cyclic molecules can convert back and forth to each other, and there's an equilibrium that forms between them as they pop open to form the linear recyclize. This reaction is happening all the time. But it turns out that the beta glucopyranose is more stable, and that's because this alcohol group in the cyclic form is in the equatorial position. Now, when we think about cyclic glucose, it's a 6-membered ring, and so we do need to think of it as a chair with axial and equatorial positions. And in the alpha glucopyranose, the alcohol group's in the axial position. In the beta, it's in the equatorial. And hopefully you remember from Chem 240 that we'd rather have substituents in the equatorial position to reduce steric strain. Okay, so why do we call them pyranose, furanose? Well, they're based on these organic molecules that are 5 or 6-membered rings. And so when we think about cyclic glucose, we can call it glucopyranose in either the alpha or the beta version, where this, what we're going to call the anomeric carbon, differs in where the alcohol substituent is. So glucose, carbon number 1, is what we're going to call the anomeric carbon, A-N-O-M-E-R-I-C. That's because the alcohol group at that carbon can differ. And here in this particular projection, we see it's either up or down to define the alpha or the beta. Okay, so again, we need to think of these as chairs, and here's the beta form of cyclized glucose with this anomeric alcohol group in the equatorial position. So that is a talk about monosaccharides like glucose and how they can cyclize to form alpha or beta versions. The next video is going to talk about how we tie these monosaccharides together into disaccharides and polysaccharides.